with our celebration of US HUPO awardees. And it gives me great pleasure to introduce to you Catherine Costello. Kathy is a professor at the Boston University School of Medicine, where she established the Center for Biomedical Mass Spectrometry. And I think that it's perhaps not surprising that anyone who gets to know Kathy becomes instantaneously a fan and a friend of Kathy. And the reason for this is that Kathy embodies the representation of creativity, rigorous science, and leadership. Kathy was the president of ASMS and the president of HUPO, and she currently is the president of International Mass Spectrometry Foundation. And she has impacted numerous areas of science, but her primary scientific love and focus has been on glycoscience mass spectrometry. And she has been at the forefront of this field for a number of years. And in recognition for her contributions to science and her legacy, US HUPO last year named the Lifetime Achievement Award in Proteomics after Catherine Costello. So the award is now named Catherine Costello Lifetime Achievement Award in Proteomics. And this is the first award in mass spectrometry or proteomics named after a woman in science. So congratulations to Kathy. The recipient this year of this award is Professor Rudy Abersold, who will talk next week on April 22nd. So tune in for his talk. But we have now invited Kathy to give an overview of her career and her feeling of where proteomics has been and where it's going ahead. So without further ado, it gives me great pleasure to welcome Kathy to the stage and give her, and hopefully we have the slides ready for Kathy.
Thank you very much, Ileana. It's really a great pleasure to be here. It's unfortunate the circumstance that brings us together in this way, but my best wishes to all of those who are affected. But it will be a pleasure for me to give a short summary of the major milestones and how I came to be working in proteomics and how it's driven both by the people in the field surrounding that I've been lucky to work with or collaborate with or know or benefit from their experience. And also along the way as I'll illustrate the development, the continuous development of instrumentation and methodologies that go along with them that make it possible to reach much farther than we could possibly think about when I was first starting to do this. So I put this first picture on here is just this endless tunnel, but with some light in it all the way. And that's kind of how I feel that the course has been. So I started as a PhD student in organic chemistry at Georgetown University in Washington, DC. And it was a classical physical organic chemistry program, or the project that I took on, which was to figure out if in bromonium ions they're actually symmetrical or not, and if not, what things affect that. So I used a lot of infrared spectroscopy, UV vis, ORD, CD, a lot of very low temperature NMR to control changing the temperature and see how that changed the stability. Along the way, because of the location there, I had the chance to do some internships at the Food and Drug Administration and the US Department of Agriculture. But I was really interested in chemistry to try to make some impact on the life sciences. And I gradually was able to work on that. And because Georgetown is very close to NIH, we were fortunate to have a number of the investigators from NIH as our teachers in our courses. But as you see, there's no mass spectrometry listed here. And in fact, there's only one mass spectrum in my thesis, and it was run by someone else. But I did get an exposure to it because I wanted to get to know people from other countries. And one way to do that was to go to the NATO summer courses. And as a post-starving graduate student, that's how you can travel. But it was a course on mass spectrometry. And after being locked up with people for 3 weeks, experts in the field, and getting to know I found out what's going on and how many advantages it would have over some of the techniques or in complement to them that I had been using. And the suggestion, you go to Scotland and you find people who suggest that the good place to go for a postdoc was back to Boston, where I grew up and where I went to as an undergraduate. But somebody was really getting going at MIT who was showing how much structural information you could get by mass spectrometry, and in fact was starting to sequence peptides at least, and showing that these kinds of things were feasible and lots of natural products. And so then a couple of years later, when I did graduate, I went up to MIT as a postdoc, and there's a picture of Klaus, who always looked younger than his students and postdocs. And he got very involved in the Viking project to put the small mass spectrometers on Mars, and that's the reason that it's a NASA picture of him here. But he was a wonderful teacher and someone who was very interested in where mass spectrometry could go. And the first project that I worked on, and I worked on it for several years, was to set up a system to use GCMS to do toxicology, particularly for when people, and we focused on children, were brought to the hospital emergency rooms to see if we could quickly tell what drug it was they had taken. And especially when you deal with children, there haven't been clinical trials on adult drugs done on children. And when you encounter street drugs, there haven't been clinical trials done nor metabolomic studies done. So you find a lot of things in these samples that one needs to elucidate in the middle of the night, sitting on the floor, trying to figure out quickly what the pharmacology could be. But it gives you an appreciation for what mass spectrometry can do, even in a system like you see here that was a single focusing magnetic system from Hitachi with a Perkin-Elman 990 GC interfaced by a piece of glass that had to be blown back into place each time that you wanted to clean the ion source. But it was one of the first systems that was put together with a computer, and there was activity going on all the time in the lab to improve this. And so this was a great help in developing this methodology. And we developed a reference set of spectra for drugs, drug metabolites, and normal components of the different body fluids. And these were then made finally into a chemical rubber handbook, but distributed also by all the mass spectrometry manufacturers to try to get this approach working. And one of the people who was also early in this area was Hank Fales at NIH, who had been one of my teachers at Georgetown and started doing the same kind of work there. A few years later, things looked quite different, and this is by 1985. We got funded, and this was a wonderful program that NIH has had for many years, first from the National Center for Research Resources, now handled out of imaging and general medicine, of developing research resources where they will make complex instrumentation available to laboratories who will then continuously develop this, and they will allow you to ask for something which doesn't exist. And then if it works out in place, then it will become more generally useful. And at the same time, closely coordinated with applications in the biomedical sciences. So we got this 4-sector instrument from JO on this kind of speculation, where they funded us without knowing what company could possibly build such a thing. And in the end, it was JO who built it for us. And it was 20 feet long and weighed 12 tons. Fortunately, we were in the sub-basement, but even that needed support columns. And you see there me with much more hair than people normally have now. And Steve Martin, that I think many of you know, and Tetsuko Matsuo, who was the designer of the optics, who would visit us about once a year so he could do some experiments because he didn't have one himself. But with that, it was possible to do MSMS with high energy collisions in the kilovolt range, and to separate at both ends in high resolution so that we could look at monoisotopic peaks. And this is then, and I put this as really high energy because now we talk about HCD all the time, but compared to the kilovolts of energy we're talking about here, it's actually rather low. And under these conditions, we get more sidechain fragmentation. And in this case here, you see for renin substrate, a whole series of A and D ions. But this kind of information then could easily be reproduced and could build up references and also rules about how this kind of dissociation would take place. And it was very good for proteins and greatly advanced our ability to sequence proteins and the peptides that would come from them, way better than the little short things we could do with the GCMS or the intervening instruments. But I was always the one that was working on the things other than proteins in that lab and developing interests in lipids and carbohydrates and their importance. And at that time we had as a postdoctoral fellow, Bruno Domon, and he worked closely with me to develop the rules for fragmentation of glycolipids and glycan fragmentation. And these then were adopted by the community and have been used since. And I might point out in the context of this award that in fact Bruno for a number of years later, much later on, worked in fact together with Rudy. So we share appreciation for Bruno's skills. But we want to talk where did proteomics come from. So this was really in the, what I would call the springtime. The flowers were just poking above the surface to tell us what would come. And it was the development of ionization modes that let us look at much higher molecular weight pieces that really moved this along. But the people that were there in the early spring were Klaus. Again, you see him in the picture here in the center. And then Howard Morris and Don Hunt, who worked at different times with both Klaus and Howard, and also started developing his own schemes for doing MS and MSMS to get peptides going. So the three of them really contributed very much in the early days and continued to for years afterwards, but it was the developments brought about by other people in the pictures here. And to the left on there, I guess I have an arrow. So we have Michael Karras. Oops, sorry, it went backwards. Can I go back? There we go. I won't point at them anymore. So we have on the left Michael Karras and on the right Franz Hillenkamp, who developed matrix-assisted laser desorption ionization. And then we have John Fenn in the front there, who developed in a way that became very useful for a wide community, John Fenn. But I point out on the right, Lydia Gall, who in St. Petersburg also developed this technique, but at a time when the Russian science wasn't very well known in the West. And so the growth in the development really came from John's work. And then we have Bernard Spengler, who developed the the dissociation of the ions that we could be using in a reflectron tandem mass spectrometer. Then we have, so we got early on, fortunately, again from NCRR being willing to bet on something not yet developed. They found it, they gave us the funding to buy a MALDI-TOF instrument, but again, none existed as a commercial system. And so Marvin Vestal decided that he would turn his company called Vestec into making such an instrument. And it was a design come up that Brian Chait made at Rockefeller. And so you get, if you get the first one, this is how you get a personalized delivery. And so this was pushing it past the food trucks at MIT. And then Franz sent over one of his excellent students, Ant Ingendo, who helped us get going with the system. And one of the first things to show that not only proteins would work well, but we decided to look at site-specific glycosylation. And so we could take peptides that had been digested from a very highly glycosylated protein, alpha-1 acid, and separate the fractions using lectins actually to different types, and then look at each one of these by as MALDI samples and plot out all from each of the different glycosylation sites what was the distribution of the different types of N-linked glycans. So this really moved things forward very much so. But we also, then when I moved shortly after that over to Boston University School of Medicine, because as Klaus was retiring and I was going to take over, I thought I really wanted to do all this to work on health-related problems, and it would be better to do that in the atmosphere surrounded by people who were day-to-day looking at diseases and what was going on, to know where the problems were that needed solving. And people thought it was kind of crazy to voluntarily leave MIT, but I've never been sad about it. It's a very collaborative climate, and it really has immersed us in direct contact with diseases. And fortunately, Finnegan Matt, who eventually became Thermo, had made a Vision 2000 reflectron TOF, and they contributed that as we were setting up the laboratory there. And we were able to do very large complexes. One doesn't usually think of doing these by MALDI, but this is an infrared MALDI mass spectrum of the intact collagen trimer that weighs about 300,000. And you see here the peaks with different charge states. It looks more like an electrospray spectrum, but for high mass compounds, and particularly with infrared MALDI, one does see these same types of patterns. So in each case, we're learning more about the technique as well as finding new ways to solve complex problems. But when we, then one of the things I wanted was to get a Fourier transform ICR instrument so we could do MSMS to the N to be able to fragment particularly glycans into enough stages that ultimately, instead of just cleavage between the sugar rings, we would also get cleavage across the rings at the energies available in such an instrument. But what we found out was that if we generated the ions by MALDI, even though it's a low energy, so-called soft energy process, there was a lot of metastable fragmentation. And so by the time the ions would be transferred from the spot where they were ionized all the way over to the cell, we would have lost labile groups like sialic acid or fucose residues, or in the case of peptides, phosphates. And Pete O'Connor was in the lab at the time, and he helped with or designed, and we had a very good shop at BU that can build many things, a design that allowed us to put in a very light pulse of gas just before the laser pulse hit. And then the ions would come off at slightly higher pressure and be able to get rid of their excess energy by interacting with the gas. So not to fragment them, but just to cool them. We called it at first high pressure, but the physicists said at that pressure, it's still not high. So we talked about what the mechanism was. So it's vibrational cooling. And this actually helped a lot. So you can see an example here, like where the three purple squares are sialic acid that are very labile groups because the carboxyl is right beside on the same carbon as the glycosidic linkage. Under normal conditions, we would see no molecular ion, but we would see fragments that come by loss of sialic acid. Under these vibrationally cool conditions, we see an intact molecular ion and only very low abundance peaks that correspond to any loss of sialic acid, which in fact could be that there was in fact in the sample a small amount. But at any rate, it's greatly reduced. So this enables us to look at labile modifications. Along about this time, NHLBI, the Heart, Lung, and Blood Institute, decided to set up a proteomics program to encourage people to develop proteomics methods that would support their areas of investigation. And they had this in two contract offerings that had a small gap between them. But we were fortunate to be one of the labs funded in both of these, so it extended approximately from 2002 to 2015. And our interest was, particularly since there's a great deal of skill and expertise at Boston University School of Medicine in cardiovascular diseases, to look at cardiovascular proteomics and particularly oxidative post-translational modifications. So I don't have time in this brief study to do that, but this is how that developed and what really pulled me back, having been the one who turned it around. So we put glycans as the upper intent for the BU lab and proteins and peptides second. We were suddenly back with proteins being a very large part of our actions. And you can see that many of the people that are still in the field, I then got into the US HUPO and the Human Proteome Organization. And by 2012, I was ending up being president of that. And we had the international meeting in Boston. And I think you can see many people that you still recognize as being active in the field and have been very important for US HUPO. Ralph Bradshaw standing beside me, Gil Omen's over on the right, Roman Zubarev, who we've already heard a bit about him, and I'm sure we're going to hear more. And the others have all taken turns being president of HUPO as well. But we still wanted to be looking along with these because more than 60% of proteins are glycoproteins. And in all of these different diseases, we want to keep optimizing the methods so that we can deal with the glycoproteins as well as proteins. And if we use ECD, we just saw Roman, who was at Fred McLafferty's lab when they developed that. And we can see that that will fragment a peptide that's a glycopeptide and leave intact the glycan. Whereas if we use IRMPD, which is available on the same instrument, we will fragment the glycan and not the peptide. So these kinds of techniques are quite complementary to one another. And I've never been one to be all in favor of any one thing or another. With the answer, the reason we want to do these things is to solve the problem. So to see what the different strengths are of the different methods, and then with each of them, develop rules for how they'll behave. Eventually, we got the Bruker-Solaric system, and then we could have a higher magnetic field, higher sensitivity, and more options for control over the fragmentation. And first with Pete and then with Cheng Lin, we explored the higher energy fragmentation. So this is electronic excitation dissociation of 14 EV, and an archaic glycopeptide that doesn't have the internal chitobiose core that is on most of the mammalian proteins, but is typical of what happens in strata like archaea and bacteria. So we can't release it, we can't use the normal fragmentation, and under usual conditions, all we see is some losses of parts of the glycan, but we don't know where they are. But with this higher energy EED, we can finally fragment the peptide, but leave the glycan intact. And in this case, we got 38 unique glycosylation sites. We can, when we just look at the glycan by itself, then induce fragmentation. And here we have electron transfer dissociation. And Liang Han, who was a student who started her work just as we got the Bruker ion trap that was equipped for this, put her on this as her thesis project. And she did a wonderful job to define how much information we can get from electron transfer dissociation, both about the sequences and the linkage positions. Then we also have to be prepared for the fact that many of these organisms do things that we couldn't always predict, and we have to keep an open mind. And here's an example of Toxoplasma gondii, which is an infective organism that particularly hurts immunocompromised people. And here we found, surprisingly, a series of glycoproteins that were not on the surface where we would expect them, but they were located right around the nucleus, around the nuclear pore. And they all had fucose on them. We were able, by different fragmentation methods, to sequence these and locate where these many fucose residues were. But in the end, what we end up with is a structure, and that has to be created by the fact that these organisms have hijacked genes from plants with which they live in the soil. And so they're producing things that their own genome wouldn't explain, but by their association with others, they can make these unusual types of structures, and in this case have these very unusual nuclear pore complexes. So one must always keep one's mind open about what can go on, and more we're appreciating what the microbiome and the impact it has on our health and on the structures that are living inside us. We have looked a lot to develop methodologies for looking at the glycosylation on the immunoglobulins. They have the IgG1, which is the most abundant, has just one glycosylation site. And the presence of variations in the carbohydrate structure there can go completely from pro-inflammatory to anti-inflammatory. This is important for places, the industries that are developing antibody-based drugs. Our interest was also to look at it at the level of the normal plasma proteins, and not only IgG, the different classes of IgG, but also IgA and IgM. And can we do this by mass spectrometry? And Kevin Chandler was the person in our group who worked on it. We did it in collaboration with Gilit Alter at the Ragone Institute. And we could, there's just a few of them here, we can see the different types of structures at various places on the IG1, 3, 4, M, and M has many sites, and some of them are high mannose and some of them are complex glycans. A also is heavily glycosylated with both O and A-linked, O and N-linked ones. So we want to find as much information as possible, and sometimes just the location can't be fully determined, as I mentioned with the archaea situation, by HCD, because we will fragment the glycan under the conditions used for HCD. But if we use ETHCD, then with the electron transfer step, we're fragmenting the peptide backbone, but leaving the glycan in place, as I showed you. And the HCD part is really not to fragment it further, but just to tickle it enough so that the non-covalent association of the fragments which were formed in the first step will be broken, and we can then see those individual fragments and get the detailed structure. And here's an example from IgE, or the immunoglobulin that has a lot to do with asthma, which is again a very highly glycosylated one. And here you see sigma fragments that give us the definition of the peptide, but also there's a high mannose structure on the N-terminus, and all those fragments that contain the N-terminus have the stars on them, and they all have the complete N-glycan structure. So now we have a much better way of looking at carbohydrate site-specific localization. But
Kathy, sorry to interrupt. We are running out of time. So
okay. I just got about 2 more slides.
Okay, perfect.
Just one other thing that we can do. We mentioned, I've already had cross-linking mentioned. We can develop, not only use the structure kinds that are available, but ones that answer specific problems. And in this case, we were looking at the prion protein, the flexible region, and the interaction with the more rigid region, where there are very few lysines. So we made a new type of reagent, and that will use a carbene to do the insertion in any bond. And so we could locate all these sites and show with low energy that we just get the peptides attached by the group, and with higher energy fragment that so that we can then get the sequence of the two peptides and know the full detail and map out under different modifications of the structure what these relationships are. So in summary, we've come a great distance, evolving rapidly, costs more as we go, but we get a lot for what we pay. We can handle things with different kinds of modifications. We need complementary techniques. We can look at things using all these different methods and get complex structures. And we need very much the people, and we'll honor some more of them here, making databases, user-friendly software, and modeling. So I thank all of you, my colleagues, quickly, the ones who have been part of our faculty, students and postdocs, a very small number of the collaborators. But thanks to HUPO, because in US HUPO, they teach us to do things that we never thought we could and play roles we can't. Here at the Geneva meeting, we turned into clowns, but time moves on. And finally, I want to leave the focus on the new Lifetime Achievement Award for Rudy Aebersold, and I hope everyone will join next week when Rudy gives his talk. So thank you all again very much.
Kathy, thank you so much for this wonderful talk, and congratulations again for this amazing and well-deserved recognition. Thank you for also your generosity in immediately saying yes to our invitation and giving your time to this webinar. So one quick question that I have for you is, just to bring this back to the circumstance that we are all facing right now and the time that we are going through. The coronavirus, the SARS-CoV-2, as you know, is an envelope virus. It has this spike glycoprotein that is essential for its entry into host cells. It also remodels the endoplasmic reticulum, the ER, in order to replicate and form these double membrane vesicles that are critical for its replication. So these compartments that are so important for glycosylations and anchoring glycosylation. So what is your thinking about the relevance of protein glycosylation during coronavirus infection?
Oh, it's extremely important because not only the spike protein is a heavily glycosylated glycoprotein, but all of the candidate receptors for it, not just the ACE2, but others as well, they're all glycoproteins, and much of the interaction that helps to position the spike protein to enter the cell has to do with carbohydrate-carbohydrate interactions. And so the interest in defining these, and of course, it's hijacking the system of the host to produce these. So the glycosylation is host-specific and tissue-specific and state-specific. So it's not that the virus itself is changing its sequence so much like some of the others, but it changes these interactions depending on who the host is and where it's located and over time. So we're very interested in getting involved in looking at this glycosylation as are other groups. And I think for the community, it's good to know that there is a consortium being developed that now has more than 40 different labs in 15 countries working together and sharing data so that we can make the most of what we can do with mass spectrometry to try to attack this problem together.
Yes, thank you, Kathy, and thank you and John Yates and all the others who have sent information about this consortium that I think it's important and it shows kind of the desire of the scientific community to step in and really help out with anything they can. So with that, I'm gonna pass the mic to the next chair, to Dave Arnott, to chair the next talks that will be from the exhibitors.
Okay, thank you. In this next portion, we've invited two of our exhibitors to give short talks. If our conference in Seattle had gone off as planned, they would have been present the whole time to talk to any of the other attendees and share information on their companies. Since that wasn't possible, we're giving them a chance to give a short video presentation to highlight, some aspect of what their companies can do. There will be some time for questions and answers, so please use the Q&A function on your screen. But I'd encourage anybody who has an interest in these companies and their technologies to contact them directly. I'm sure they will be happy to give you all